Welcome to A-level biology. I'm going to try and cover all the exam boards, so it's not going to matter whether you're going to do perhaps OCR biology, whether you're going to cover AQA biology, or whether you're going to try and get through the EdXL sort of biology. Less in that book.
Biology is the natural science that studies life and living organisms.
Yeah, very helpful as my Alexa decides to try and help me there. Whether you're going to look at biology with the EdXL, which is looking more at how biology works, and you sort of have to pick up some of the biology. So there we've got some of that. Even some of the stuff that I'm going to cover is going to be relevant if you're doing things like the BTEC Applied Science. So where I'm going to start is having a look at protein synthesis. I want to have a look at basically getting everything from DNA to the protein, having a look at the different types of amino acids, and then going on and looking at the primary structure, the secondary structure, tertiary structure, and we'll probably talk about quaternary as well, and the basic properties of proteins. So hopefully this will cover bits and pieces for all the different syllabuses and all the topics. So there we are, there's the plan. Let's have a go and get into what we're going to have a look at. So where we're starting from here is we're starting from some DNA, and we need to have a look at some properties of DNA and how it works. So let's start there, and what we're going to start with is a model. And here I've got a little model of DNA. It's got a backbone and it's got bases attached to the backbone. Let's look at that differently. Here is, if you like, the coil of DNA, and it seems to have a large sort of groove in it, and as I twist it you can see my hands going up the large groove, and it's got a much smaller groove. Okay, let's have a little look about what this really looks like and how it goes together. So our DNA consists of two basic backbones. The backbone here is made of a sugar and a phosphate. And it looks perhaps something like this. I've got my sugar and a phosphate, and the phosphate joins to another sugar, and then another phosphate, and then another sugar. Attached to this sugar is a base, and we have one of four bases available to us. We've got A, adenosine, T, thymine, G, guanine, C, cytosine. A bonds with T on the other side. T surprisingly bonds with A on the other side. G bonds with C, and C bonds with G. Now I've tried to make a little difference here, showing there are 2 bonds with these and 3 bonds with those. And that's in fact why the A can only bond with T and G can only bond with C, because it's the number of bonds, and it's how the bases actually fit together. And I can give you an idea about that, having a look at perhaps a couple of these bases. So let's bring in a couple of these bases, and I've got here a couple of the bases, and you can see they've got 3 bonds and they will in fact join together. Whereas if I look at the other combination, then I've got 2 bonds, and the 2 bonds don't fit together with the 3 bonds, so they won't basically bind together. So here's our basic structure, and let's move on from here to look at why DNA is good. Well, DNA can replicate, and if I take a couple of strands and I break them open, so we'll have our A with our T, and then we'll have a T goes with an A G with a C with a G. Right, let's repeat that pattern here. A, T, C, G, A, T, C, G, and we'll repeat this pattern. T, A, I did it wrong, didn't I? Oh well. G, C, and then T, A, G, and C. They would fit anyway. When the DNA is remade here, then this is going to produce a C with that one, this is going to produce a G with this one, an A with this one, and a T. This one's going to produce an A with a T, with a C, with a G. And we've got 2 identical chains forming here. This is semiconservative replication. And it was necessary for people to perform an experiment to try and prove that this was correct. If you want to look at that one, you can do that by yourselves. I'm going to carry on looking at how this works. So if we go back in time, billions of years, these genes are copied because the DNA makes up the genes, but just with these 4 bases. Now, why 4 bases? Well, if we put our 4 bases in sort of context with one another, I can get combinations such as AAA, AAT, AAC, AAG, and I can go through a whole series of different ones here. This will give me 64 different combinations, and we have 20 amino acids to try and make. These are the 20 amino acids that are made and available in the body. So this is sufficient. 2 base pairs would only give me 16 combinations. 16 would not be enough, but here we've got some. So it may be that these 2 could code for the same amino acid. And you can look up this chart to see which ones do which. You don't have to memorize it. you merely need to know about it. There are certain sequences, though, that mean different things. There is a start sequence, something that says this is the start. And all proteins always begin with methionine. And there is a stop sequence, which doesn't code for any amino acid, and that simply stops the gene. It just says this is the end, and this protocol is followed by all living organisms. And this is some evidence that DNA and evolution are all tied in together, very simply because all organisms, be they bacteria, that dreaded COVID-19 virus, all plants, all animals, they all use exactly the same DNA sequence. DNA codes for the same amino acids. Let's have a look and see how this is achieved. Right, so let's look at an idea. What we're going to do is we're going to take a piece of DNA, and this DNA, what I'm going to do with it is I'm going to split it. We've got, therefore, if you like, one side which is going to be the master side, which is going to be the coding for the gene that we want to look for. And the other side here is just a complementary strand which just makes sure that this gene, this piece of DNA, is going to be valid. And we've got a point to another point which is our gene. If I look at what a gene might look like, it's going to have some sort of binding site. And this binding site is going to be for the enzyme to turn this DNA into mRNA. And you can look at this with something like the lac operon. There is a very good simulation of this done by fet.colorado.edu. On their website they've got lots of simulations, and I'll put a link at the bottom to help you. We've also got another site on many genes, and this can be for a promoter stroke inhibitor. We can have this site to try and promote the gene, and we can have this site here to actually inhibit. And basically, if I can block this with an inhibitor, then my RNA polymerase, our enzyme complex that's going to go and code for the actual gene, isn't going to be able to get through. Only if this is unblocked, then will it be able to go through and produce the gene. So this enzyme is a complex, and there are some really good simulations of this, which I'll put some more links at the bottom for you. Right, let's have a look and see what's going to happen. This DNA is going to be set open somehow. This is all the process of epigenetics, of basically having proteins binding to the DNA, and if these proteins aren't bound to the DNA, then it can be copied, and if it's got these proteins hiding the DNA, then it can't. Let's give myself a little piece of DNA. I haven't got the most imaginative piece of DNA here, but we've got something. I've got here 3 sets to code for 3 different amino acids. It's unzippered, and what happens is this RNA polymerase comes along, and for every A, instead of producing an A, it's going to make a different amino acid, U, because this is going to be RNA. So this is our DNA, which acts as our template for the gene, and this is our piece of mRNA. For the T, we're going to get A. The C, we're going to get G. G, we're going to get C. So we've just got here our thymine replaced with a very, very similar looking base uracil. And there is my mRNA. In bacteria, this is the process that goes ahead. In eukaryotic cells, then it might be slightly different. We might end up with a long piece of mRNA, and then we're going to have some chopped bits chopped out.
Paul Evans would like to talk.
Right, I'll stop that for a minute. Right, okay, so we can chop out bits of our mRNA, and then we can stitch them together. Now, exactly why this happens, I don't think anyone can tell you. We know it does. So bacteria take its mRNA and they keep it. Eukaryotic cells, we take a much longer piece of mRNA and we chop out some bits and we stitch them back together again. As for the whys, I can't tell you. Okay, so let's look at this with a bit of a model. So my model of my DNA is unzippered somewhere along here. So if we start to do that, we can break this apart, unzipper it, and as we unzipper it, we can start to make our mRNA from this. So let me just put this one in there, and you can see that it starts to form. Right, would do if it would go in. It starts to form this mRNA. So let's have that, and I've now made a copy, albeit a reverse copy, of my piece of DNA, and here is my mRNA copy of that. Okay, so here I've got my gene sequence. This mRNA now leaves the cell. So we've got our cell, we leave our nucleus in the cell. So we've got our DNA here, and our mRNA travels outside into the cytoplasm, and typically round here we might have a piece of what we'll call rough endoplasmic reticulum. It's called rough endoplasmic reticulum because on it we've got lots of ribosomes. Okay, so I've got my endoplasmic reticulum, and this mRNA binds to what looks like a cottage loaf structure of this ribosome. So the ribosome consists of two bits, a smaller bit and a larger bit, and this piece of mRNA fits in there, and it goes in 3 bases at a time. Let's look at what's happening then. Well, in the body we've got, in this sort of cytoplasm, we've got lots of different other molecules, and these molecules are of tRNA. Let's just zoom out here a little bit. So I've got molecules of tRNA. This is transfer RNA. And one of these molecules will have 3 complementary bases on it. They're complementary to this mRNA. Connected to these, there is a specific amino acid. So there will be 20 different these called tRNA, and its structure is here accentuated in this model, but in fact it doesn't look like that. So let's see what it does look like. We have a molecule that looks something akin to this. Down here I've got my 3 bases, and these 3 bases are my anticodon. It's opposite to the 3 base pairs that we've got on our mRNA, and that was the opposite to what we had on the DNA. So we had 3 pairs, we've got the inverse on here, and then we've got the inverse of this on the tRNA. So I've got these 3 bases. This sequence, we've got 64 of these different ones, or thereabouts, and attached to this is a specific amino acid. And this is my model of this. So I've got my amino acid, I've got this large structure, and it's rather distorted in this case because I've got my 3 base pairs. So we go into the ribosome, and let's have a look and see what's going to happen here. Let's move that a little bit out of your way, and we might be able to see what's going on. So I've got these different bases, and we want to see what's going to happen. So the first one's going to come in, and we've got our complementary strand. So my dark blue one here is going to correspond to my light blue. My yellow is going to correspond to my green. Get it in nicely, there we go. And my green is going to correspond to my yellow. So we've got these. I've just managed to break my model. There we are. Going in. Now this is bonded in the cell, in this ribosome, and then it shunts along 3, and another one of these comes along, and let's just get this to group in. So my yellow is going to combine with this one, my blue with my light blue, my light blue with my dark blue, and they're brought so that these two amino acids are actually brought very close to one another, so close that they're actually fused together, and this one breaks off this piece of tRNA. As we shunt along one more, this first base is now released back into the cytoplasm, and as it goes into the cytoplasm it can then pick up another amino acid. We've got this one shunted along a little bit more, and now we can pick up another base sequence. So here I've got this one, I've got my green going with my yellow, sorry my hands get in the way, I've got my light blue with my dark blue, my model has fallen to bits, and my green with my yellow. Guanine and cytosine, these. And it's brought close together. So this now fuses on, and this breaks off. And it shunts along again. Ready for our final, in this case, that's all I've got in my sequence, my amino acid to be formed into this chain. We've got these base pairs, this codon and anticodon joining together, putting in another link to my protein chain, breaking that one off, and as that goes off that's going to fall off, and eventually out's going to come my protein. So we've got here some sort of protein made simply by having these bases connect, release, connect, release, and as they do so they're joining on these proteins. Right, so we've got my particular sequence here set up. Okay, I'll just put that one back on, so when I want to use this the next time, it's all ready to go. Right. So I've got my tRNA and my mRNA back as they were. Okay, so these molecules freely made inside the cell, we've got them in the nucleus, and they will have nucleotides in the nucleus. So we've got in the nucleus, let's zoom in a bit for you, when we're doing DNA synthesis, we've got lots of phosphate joined to a sugar, joined to a base. We've got lots of these little things available. Now for DNA, the sugar will be deoxyribose. For RNA, then we've got this phosphate and sugar with its base, but the sugar here is going to be just ordinary ribose. What's the difference? Well, the deoxyribose changes the shape very, very slightly. So the sugar here is going to enable a straight chain. And this is going to form more of a helical chain. This is going to allow this to become double-stranded, but this being straight is going to make this single-stranded. So we find that our RNA is always a single strand, we find our DNA is always a helical strand, and it's always, in fact, formed as a double helical strand. We don't find much single-stranded DNA available in nature. We do find, if we're trying to do protein synthesis and various other things, DNA replication, when we're trying things like PCR machines, which are used widely at the moment to try and sort this COVID thing out. Right, the tRNA is a bit of an oddball. The tRNA is largely single-stranded. However, it does have some double-stranded parts. So our tRNA is the same. We've got basically the same structure here, it's just some of it's allowed to be combined. Now, if we look at our DNA, we've got the bases of A, T, C and G, adenine, thymine, cytosine and guanine, and in the RNA we've got A, U, C and G, adenine, uracil, cytosine and guanine. So there is our DNA replication, with our RNA being produced. There is our RNA being copied into proteins. So we've got two basic processes going on. We've got a process of transcription. This is when we get DNA copied to DNA, and when we have DNA copied to RNA. And then we have the process of translation, when our RNA is changed into a protein. And I want to have a quick look now at this protein. What does one of these proteins look like? Let's zoom out a bit, wrong way. So I'm going to make a little molecule of an amino acid, and I'm going to use these snap-ons to do so. Not particularly good. Let's move these out of the way. I don't have enough space to work here. Right, it's an amino acid. So let's start with that bit. So we've got an amino group, which is NH2. This is connected to a couple of carbons. The second of the carbons is the acid, so that's got, looking at this not being very helpful, you're not seeing it in the right place. Let's put in a double bond here to an oxygen. And we've got an OH. And this basically is my amino acid. I've got an amino group, one end. I've got an acid group at the other end. I've got a hydrogen onto a carbon, and then this other group can represent a whole series of different things that can be put on there. The simplest of these would be just putting in one hydrogen glycine. A more complicated version might be to put in alanine, which is going to be a CH3 group. I'm going to build myself another one of these so we can play with it. So what I've got is my amino group. We've got the two carbons. We've got a double bond to an oxygen. And then we've got an OH group. So there is my carboxylic acid group, my acid group. On this side we've got the middle carbon, and we'll shove on there alanine. Right, having tidied up a little bit, let's see what's happening inside the ribosome. Inside the ribosome I've got these proteins. We've just brought in one of these, and we bring the other one very close to this. And as we bring this in very close, then something happens. We lose a hydrogen off this one, we lose the oxygen and hydrogen off that one, and these two now join. We've got water formed. It's a condensation reaction, and now I've got two amino acids next to one another, and I have this peptide bond formed. If I look at what this peptide bond is, it's a carbon with a double bonded oxygen on it and a nitrogen. Let's just draw this out to see what's going on. Right. I'll zoom in for you. That'll make your life easier to see. And we've got this NH. We've got a carbon with a hydrogen on it, and any other particular group. There's a whole series of different 20 of these, and we've got our carbon with a C double bond O and an OH group. So here is our basic amino acid structure. Let's join two of these together. We're going to make a dipeptide. So these two are going to come close to one another. This is going to be removed. Here is our peptide bond formed. And I'll just leave the rest alone. So we've got here a condensation reaction formed because we've got water made, and this here is our peptide bond. This goes on and on and on in proteins, so I start joining all my different amino acids together. So we can join them in any random sort of group, and this makes our primary structure. This is simply the order of the amino acids. Now, if we look at some of these these amino acids, we can get some idea about what's happening. If I've got two glycines next to one another, they've only got small sort of groups, and they can sort of, they could only have sort of certain interactions with one another. Let me zoom out, you might be able to see better what I'm doing here. They got certain positions they can go to. My molecules are still falling to bits even though I'm using the new snatoms. But we can get different sorts of here amino acids. We can get different ones. So let's just see if we can make a different one of these and see what we've got. So I've got glycine, I've got alanine, but let's have a look and see what we can do with these. Now, I've got a list of these in one of these BTEC books of some of the different protein amino acids, so we'll have a look at one of these. And yeah, it's typical, I'm going to lose the page. I had some nice pages of the different, here we are, I found it. Now, we've got all sorts of different chains that we can put on, and if I look at one of these, and I'm going to make lysine, we can make some interesting observations. So if I'm going to make lysine, I'm going to have CH2, 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 CH2 and an NH3 on there. So let's see if I can just put that in. So I want my NH2, and then I'm going to want a 4-carbon chain with its hydrogens in place. There we are, there's my molecule. And what I'm going to do is I'm going to try and put that back with my other one. So I'm going to take my lysine, take it off. I'm going to put on here. Here is lysine. Now, let's see what's going on. I've now got an enormous sort of molecule. Let's look at what we've got. I'm just going to break this. This is alanine. Quite a reasonable amino acid here. It's got its carboxylic acid, it's got its amino group, and it's got just one methyl group attached. Lysine, on the other hand, here, the same basic structure. We've got my carboxylic acid, we've got my amino group, we've got my carbon with its hydrogen on it, and then I've got this big long chain sticking along. Let's get this to react with the other one. The water is formed and these two join together. Now we get a problem of something called, if you like, steric hindrance. This can move in many ways, this can move in many ways, but these groups are actually quite big and they can interfere with one another. They get in the way, and so these molecules tend to orientate themselves so they can only fit in one pattern. If they can only fit in one pattern, then these molecules can only be arranged in one particular way. So if I move that out of the way, and we come back, we've got then this primary structure. We've then got this secondary structure, which is formed by basically how big the side chains are, how close those side chains can go to one another. Just focus it for you. And this determines the shape of the molecule. So now we've got an idea about how our amino acid sequence can modify the shape of the molecule. So if we've got ones that are close together, we can get twists. If we've got big lumps, then they're going to make particular angles. So we can get a molecule that might be a particular shape because we've got a nice bit that can make a coil and another bit that might make a coil, but then we might have two big amino acids with chains sticking out here and there. That means that it can have to bend around. If this amino acid gets changed for any reason, then this is where the whole shape of the molecule could be changed. And this is one of our mutations. That will happen because of just one base necessarily being changed in our DNA. If I can bring this chain close by to that chain, then maybe I can put a bond across there. And we see with when we made our lysine had an NH2 group on it, had a chain coming out with an H2. This added an extra amino group, and maybe that could hydrogen bond with another group, especially if we had an acidic one. And that causes our tertiary structure. Right, so to finish, let's look at how this might happen. I've got my DNA sequence. Now, what would happen if, for some reason, this base could get changed? Now, this happens all the time, but if we look at our double helix, if I remove this group here, then the self-repairing system, so let's just try and remove it, the self-repairing system would come along and say, ah, look, you've got a T here, so I would have to replace this with an A. Maybe something happens to my DNA, and for some reason we can change this, and I can change this for any one of an A, T, G or C can go in there, and this will make one amino acid, and this one will make now a different amino acid, and we've got a change. Our final option would be that this sequence, something happens and this one is lost. So my next one comes along and is just the same as it was before. This is my original amino acid. This is now a new one, and this one, everything has been shifted along one. And this tends to make rubbish for the rest, and that's totally destroying the protein structure just by omitting one base. And in fact the same is true if we put an extra base in. So we can swap one, and that can change, doesn't necessarily will, change the amino acid. And if it happens at an important place, like a bend, then this is a mutation, this is going to be a significant change. If, however, it happens somewhere else and it might not be significant, then we can just end up with a variant which is not so problematic. There we are, some ideas on DNA structure and leading on to sort of protein synthesis. I hope that helps, and if you do, then please subscribe to some more. I'll see you next time. Bye bye.
Here's your reminder. Take a Puritan tablets.